Hi everyone, this is the Plant Pigment Lab using highlight and low light soybeans. For this, you will have your soybean plant, weigh scale, and you will have 2 sets of test tubes. You will have 1 set for highlight plants and 1 set for low light plants. And for each of these, you should have, again, another set of test tubes. The first set of test tubes is for the coarse slurry, and then the second set of test tube is for the filtered pigment solution. You will also be using a spectrophotometer, some 80% acetone, and some quartz cuvettes to do your measurements later. And you will also have an ice bucket with a lid to keep your solutions in the cold and in the dark. This is important because warm temperatures and light will cause the plant pigments to degrade faster. So you want to keep everything cold and in the dark. So for this demonstration, I'm only going to do 1 sample, but the procedure is the same for all 8 of your samples. First thing you want to do is select your leaf. So here I've already selected a fairly large leaf. You should talk to your TA about which set of leaves you should be using for this lab. And you want to make sure that the leaves are all about the same age. So you want to remove the large veins in the leaves. But before we do that, you want to set up your scale. I've turned on my scale already. We can take the lid off and then just put your weigh boat and then 0 it. Set it to 0 and then we can start. So using your razor blade, be careful with the razor blade because it is quite sharp. You want to cut out all of these large veins. You want only just the leafy parts in between the large veins. So just like that. And then you want to cut your leaf up into small pieces. And oops, you know what? I'm just going to re-zero that. Oh, it's already zeroed. Okay. And you want to weigh out approximately 120 to 130 milligrams. Okay, so here we have about 128, if you like. If it's windy in the room because of the air vents, you could put the lid on, let it settle down, and wait for the scale to stop moving. So here you can see we've got 0.131 grams or 131 milligrams. So record your weight for this sample, and then you can move on to grinding. So once you've recorded that weight, you can take the lid off, take your leaves out, and now we're going to grind. Once you've weighed out and recorded your leaf weight, you can take your leaf samples, bits, and transfer it to the mortar. From here, you want to add a tipful of sodium bicarbonate. So take your spatula and just get a tipful, add it to the mortar. And then you want to add 2 milliliters of 80% acetone. So I've got my pipetter already set to 1 mil, and I will use it twice. So that's 1. And 2. Make sure you put the cap back on to the 80% acetone bottle because it will evaporate fairly quickly. Just take your pestle and you want to start grinding your leaf samples. You want to make sure that your leaf samples are very well ground. If you still see pieces of leaves, as you can see in here, you're not done yet. You want to make sure that there are no distinguishable pieces of leaves left. If any of your leaves get stuck on the side of the mortar, just use your pestle and push them back down into the acetone. So you can see here, I've got this green acetone solution, so the pigments have been released from the leaf sample, and you can see I don't see any large pieces or small pieces of leaves now, so you're pretty much ready to go. But if you're in doubt, feel free to just do it just a little bit more. Okay, I think that's good. So now you want to take another 2 milliliters of your 80% acetone, and you want to get your sample tube ready with your funnel. Move this out of the way. Move that up. Okay. So I usually like to take the first milliliter of acetone, and I just like to clean off the pestle. So just clean all the solvent off the pestle, and you can put it in there. So take another mill, and you want to just wash off the sides of your pestle or mortar. Give it a bit of a swirl, and then you can just pour it into your sample tube. You can take another mill of your acetone and just rinse out your mortar. Okay. Now you want to switch your setting on your pipetter to 500 microliters. And there is a locking mechanism on some of them. Make sure you unlock it before you change it. Lock it when you're ready to use it. We'll use another half mil of acetone, and we'll just rinse out the funnel. So there might be a little bit left on the funnel tip. You can just try to brush it against the side of the tube. And now you want to put your cap in your test tube. This is very important because the acetone will evaporate if you leave it open. So now you want to take your sample and put it into your ice bin and put the lid back on. Keep it in the dark and on ice. And you want to do this for 10 minutes. At the very minimum 10 minutes. It's okay if it's in there a little bit longer, but minimum 10 minutes. And at the 5-minute mark, we will take it out, vortex it for a few seconds, put it back on ice for the remainder of the time. While you're waiting for your samples on ice for that first 10 minutes, you can go ahead and start processing your other samples. You can take your mortar and wash it out at the sink, dry it, same with your pestle, dry it, and then start with the next sample. And same with your funnel. You may have a second set to work with. If you do, great. You can just use them, but you will have to wash your equipment in between. So here we're at about the 5-minute mark. Your first sample has been in the ice for 5 minutes. We're going to take it out, just wipe off any ice, and we just want to vortex it for about 3 seconds. Take it, put it back on the ice in the dark for the remainder time. Okay, the sample has been sitting on ice for 10 minutes now. So before we take it off the ice, we're going to get ready to filter it. So take your funnel, put it into your graduated cylinder. You take a piece of filter paper, and you want to fold it so that it fits in the in the funnel. So fold it into quarters and into eighths. And again, do the same with the other side. And you just want to get your filter into your funnel. It may want to pop out, but it's okay. As soon as you pour your solution in, it will stay in place. Okay. I'll also remind you, you should be wearing gloves when you handle it, just so you don't get any of your fingerprints or oils from your hand on the filter paper. So now we'll take our sample off the ice. Okay. Wipe off any ice that's clinging to it. Feel free to give it a good whirl again. Open it up. And here's where you might want to just hold the filter paper in the funnel until you get all your solution in there. So just pour it all in. And just let it drip into the cylinder. If you notice that there's a lot of residue in your sample tube, you can take a little bit of, take a milliliter of your 80% acetone. So this was set to 500 microliters. I'm now going to set it back to 1 mil. Lock it in place. Get my tip. And I'm just gonna wash off that sample tube. And we are done with that sample tube. So wait for your solution to completely drip down into the graduated cylinder, and when there's no more solution in your funnel filter paper, you can wash the filter paper. You might find this will take a couple of minutes. Or not, because it's almost done. So you can kind of see it's almost done. So I'm going to go ahead and use another milliliter of acetone, and I'm going to rinse the filter paper as best I can. So now we eventually want to make this up to 10 mils. So if it looks like there's still more pigment on the filter paper, you can just rinse it a bit more. And let it drip. Okay, so you can see my sample is pretty much done filtering. So I'm going to take the filter paper out. Let's put it off to the side. Again, I'm going to take my 80% acetone. And you want to top your sample up to 10 milliliters. Before you take the funnel out, I find it's good to take some of that acetone and just wash your filter. Be careful not to go past 10 milliliters. Okay, we can take that out. Okay, so we still have to add a couple more mils to get up to 10 milliliters. So just take your pipetter and you can just carefully add. Okay. So now you want to take your second corresponding test tube. So this was my highlight sample number 1. So this is the second tube for the highlight sample number 1. So clean tube, take your funnel, put it in, and you just want to pour your sample into your clean tube. Shake off your funnel, put that off to the side. And then you want to cap your tube because you don't want the acid, oh, and don't forget to cap this as well. Cap your tube so it doesn't evaporate. Now you can either wrap this in foil and put it in the refrigerator, or because it's closer, we can also put it back in the ice in the dark until you processed all your other samples and you're ready to take your absorbance readings. When you're done filtering your sample in your graduated cylinder and you've transferred it to your clean test tube, you want to rinse out your graduated cylinder with some 80% acetone. So just give it a quick rinse or two. And you want to make sure you do this in between each sample.